Hello and good afternoon. We are the Group 1 and the topic for today's report is pipetting procedures or techniques. So let's get started, but before anything else, how did the pipette develop? Louis Pasteur needed a method to keep things clean and sterile during research. As a result, he developed glass pipettes, today known as Pasteur pipettes. Pipettes are essentials in almost all of the laboratory procedure. Pipettes are used for obtaining particular measurements for liquids. Lacking in knowledge about proper pipetting technique might affect your overall experiment. It should be remembered that not all glasswares are designed either to contain or to deliver, and there are different classifications of pipettes.
But before that, this is an example of Pasteur pipette.
So there are classifications of pipettes. According to manner of calibration, first is to deliver, or we called it TD. This type of calibration will dispense that exact volume indicated. The small amount left in the pipette tip should not be blown out. Water is used as the calibration medium. Second is to contain, or TC. To contain pipettes will hold or contain particular volume but does not dispense that exact volume. The exact volume can be delivered by repeated filling, then emptying the diluents, or we called it rinsing techniques. To deliver or TD pipettes are much more common than to contain or TC pipettes. Most typically graduated pipettes or bulb pipettes are usually calibrated to deliver, whereas capillary pipettes are adjusted to contain.
Are you still confused by the terms TD and TC? Accordingly, the abbreviations TC and TD stand for to contain and to deliver. Therefore, the liquid contained in a TC marked pipette corresponds to the capacity printed on the pipette. In contrast, the delivered quantity of liquid in a TD marked pipette corresponds to the capacity printed on the pipette.
Next is to blow out pipette. Similar to TD pipette, except that the exact volume is obtained when the last drop on the tip will be blown out. An etched ring or frosted ring indicates its calibration. Last is the between two marks pipettes. Exact volume is calibrated to fill the volume between two calibrated points on the pipette.
Let's watch the video of calibration and use of a volumetric pipette.
The materials that you'll need to calibrate your pipette are your pipette, deionized water, a beaker for waste, a 50 milliliter beaker, deionized water in a spray bottle, and a pipette bulb. Start by running deionized water through your pipette. Dry the outside of your pipette with a paper towel. If your pipette is clean, you should see no water droplets inside of it. If there are any water droplets inside of your pipette, this indicates that your pipette is not clean and it should be cleaned before use. Ensure that the outside of the pipette is dry so that any residual water droplets seen are not on the outside. As you can see here, I've done an inspection on my pipette, and I can see no water droplets inside of it up to the volumetric mark. In this pipette, we can see water droplets underneath the volumetric line, which would indicate that this pipette is not clean and would need to be cleaned before use. Start by weighing a clean 50 milliliter beaker on the analytical balance to the nearest milligram. When using the balance, ensure that your 50 milliliter beaker is centered. Record the mass of your 50 milliliter beaker in your lab notebook. Take water up into the pipette past the volumetric line. When taking up water into your pipette, Ensure that the tip of the pipette doesn't touch the bottom of the beaker, as the tips are very fragile. Release your pipette bulb, and using your hand without the glove, cover the top of the pipette so that water cannot escape. Carefully release water to the point where the meniscus lines up with the volumetric line. If lined up correctly, it should look like this. Release the water into the 50 milliliter beaker that you weighed before. Wait for the pipette to completely drain. This should take about 20 seconds. After your pipette has completely drained, carefully touch the tip to the side of the beaker. A small amount of water should remain in the tip of the pipette. These pipettes are specifically calibrated so that this should not affect the amount of water that you've transferred. Reweigh the 50 milliliter beaker that you've just transferred water into. Determine the mass of water that you've just transferred. Using the density of water at the temperature of the laboratory, determine the amount of water that's been transferred. Compare this value to the theoretical amount of water that should have been transferred with the pipette. Your experimental value should agree with your theoretical value within 0.003 milliliters. Repeat this procedure 3 times total. Your results for each trial should agree within 0.003 milliliters. Average these results to determine the absolute error of your pipette.
B. According to graduation. First, volumetric or transfer pipette. It transfers a known volume of liquid. It is calibrated to deliver accurately a fixed volume of a dilute aqueous solution. They include both volumetric Ostwald-Follin pipettes. They consist of cylindrical bulb joined at the both ends to the narrower glass tubing. Volumetric pipettes are designed to very accurately transfer a specific amount of solution. Volumetric pipettes are commonly used in analytical chemistry to make laboratory solutions from a base stock as well as to prepare solutions for titration. Second, measuring or graduated pipe. But first, what is a graduated pipe? A slender calibrated tube used in laboratory to measure the volume or transfer of a particular quantity of liquid from one container to another. These are calibrated to dispense different volumes. There are 2 types of graduated pipes, the Moore pipette and the serologic pipette. The Moore pipette is also known as a graduated pipette. It is a type of pipette used to measure volume of the liquid dispensed, although not as accurately as a volumetric pipette. These use a series of marked lines to indicate different volumes. Serologic pipette. The serological pipette is a laboratory instrument that transfers liquids measured in volume by ml. Most have graduations on the side for measuring the liquid being dispensed or aspirated. The use of serological pipettes is an efficient and gentle means of mixing cell suspensions. What is the difference between Moore and serological pipette? A serological pipette is a graduated pipette in which the calibration marks extend all the way to the tip, while a Moore pipette is a graduated pipette in which the calibration marks do not extend to the tip but at a point above the tip.
Let's watch the video of the volumetric and graduated pipette.
Start off using your pipette bulb to withdraw your solution into the graduated pipette. Make sure to stop at exactly the 0 point, as indicated here. Next, take it out of the solution and wipe off the excess with a Kim wipe. Place it into your desired beaker and deposit the solution. Make sure to stop at exactly the 10 milliliter mark, as any below this is excess and needs to be discarded into a waste beaker. The metric pipette is similar to the graduated pipette, but it requires all the solution inside of it at this mark. There is no excess. Start by withdrawing your solution into the volumetric pipette. Be sure to stop just above the line, as you may contaminate the inside of the pipette bulb. Stop right at this mark and wipe off the excess on the tip with a Chem wipe. You will need to drain your entire solution into your desired beaker. This type of pipette is known as a blowout pipette, which means you need to use your pipette bulb to blow out the remaining solution inside the pipette. With using either pipette, start by placing it into some soapy water and withdrawing some liquid inside of it. Next, you need to tilt it back and forth and ensure an entire coating of the inside of the pipette without spilling any liquid. Lastly, you need to rotate it towards yourself to ensure coating of all sides. Once that is finished, you need to drain this into a waste beaker. Once finished, you need to start by withdrawing some tap water inside of it and repeating the exact same procedure. Tilt it back and forth to ensure coating the pipette and rotating it towards yourself to coat all sides without spilling any liquid. This will be deposited into the exact same waste beaker. Repeat this procedure 6 to 7 times.
Micropipettes. So Heinrich Schnitger, a postdoctoral researcher at the University of Marburg in Germany, invented the micropipette in 1957. So micropipettes are utilized to transmit volumes in the microliter range accurately and precisely. And the units used for this are microliters or milliliters. In microwork, the remaining volume is left in a pipette that can cause significant error. For this reason, most micropipettes are calibrated to contain the stated volume rather than to deliver it. The fluctuation in depth during aspiration, which is dipping the dip too profoundly in the solution, can result in the droplets on the side of the tip or pressure variations that affect volume is one of the many problems or mistakes in utilizing pipettes.
Micropipettes are used to measure small volumes, usually volumes smaller than a milliliter. There are different types of micropipettes for measuring different ranges in volume. Each pipette is marked to indicate what range of volumes the pipette is used to measure. This is a P1000. It is used to measure volumes from 100 microliters to 1,000 microliters. This is a P20. It is used to measure volumes from 2 microliters to 20 microliters. This window on the pipette shows the volume of fluid that the pipette is set to take up. For this P1000, the upper digit in red represents the thousands place. The next digit down represents the hundreds place. The digit on the bottom represents the tens place. This micropipette is set to take up 950 microliters. For this P20, the top digit represents the tens place. The next digit down represents the ones place. The digit on the bottom in red represents tenths. This micropipette is set to take up 12.5 microliters. We will use the P20 micropipette to demonstrate how to use a micropipette. However, the principles learned with respect to this pipette generally apply to other types as well. Different micropipettes have different but similar mechanisms for setting the volume they will take up. To set the volume on this P20, gently pull up and turn the black wheel, as shown here. This pipette has now been set to 20 microliters. Once you have finished adjusting the pipette volume, push the black wheel back into its original position. Never adjust the pipette volume outside of the volume range it is designed to measure. This damages the micropipette. In order to take up the indicated volume of fluid using the micropipette, you must first place a disposable plastic pipette tip onto the end of the pipette. Different types of tips are used with different micropipettes. This clear tip is used with this P20. To take liquid up into the pipette, first place the tip on the micropipette, as shown here. Make sure the tip is firmly seated on the end of the micropipette. Fluid is taken up into and expelled from the pipette tip by using the micropipette plunger. The pipette plunger is operated by pushing down on the button on the top of the plunger. The plunger has two stops. To aspirate fluid up into the pipette tip, push the button down to the first stop. Continue to hold down the button as you place the pipette tip into the fluid that you want to draw up into the pipette. Only the very end of the pipette tip needs to be placed into the fluid. To draw the fluid up into the pipette, slowly release the button. Make sure that you keep the pipette tip in the fluid until the plunger has completely regained its original position. Draw the tip out of the fluid in the microcentrifuge tube. The micropipette was set for 20 microliter, so the volume of fluid taken up into the pipette tip is 20 microliter. To expel the liquid from the pipette tip, place the pipette tip into the tube in which you want to expel the liquid. Then slowly push down on the button at the top of the plunger. As you push down on the plunger, the fluid in the tip will be pushed out into the tube. Remember that the plunger has two stops, and that you push to the first stop when you are taking fluid up into the pipette tip. To remove the last bit of fluid from the pipette tip, push past this first stop to the second stop. This will expel any liquid remaining in the pipette tip. Hold down the button until the tip is no longer touching the fluid. Otherwise, you may aspirate the fluid back up into the tip.
Okay, so let's proceed for our procedure on how to properly use a pipette in a laboratory. So our first step is to wear gloves, mask, bouffant, and laboratory gown. And PPE, always remember that PPE is almost always necessary to protect workers from chemical exposures and other hazards. Second step is to prepare a proper pipette to be used. Third, rinse the pipette with a small amount of solution to remove any water film from the inside walls. Fourth, insert the pipette tip into the liquid and slowly release the pressure on the pipette bulb. Fifth, remove the bulb and quickly fit your index finger over the stem. Sixth step is to allow the level of liquid to drop until the meniscus is exactly level with the calibration mark by adjusting the pressure of the index finger. Seventh step is to touch off the hanging drop from the tip of the pipette. 8. To transfer the liquid into another container, simply release finger pressure on the pipette stem and allow the liquid to drain freely. 9th, wipe the outside part of the pipette. Touch off the drop of liquid hanging at the tip into the transferred liquid. It is a part of the delivered volume. So, we must always remember that we should never use a pipette to transfer heated solutions or incompletely dissolved solids. The heat will distort the calibration volume and solids can condense out on the colder glass walls of the pipette, clogging the narrow stem and that tip. And to better understand how to properly use a pipette in a laboratory, we also prepared looked for a short video that you can also watch.
Welcome to the video on techniques on the use of a pipette. In this video, we will show you how to deliver 10.00 mils into an Erlenmeyer flask properly using a 10 ml volumetric pipette. First, we inspect the pipette to make sure that the tip is not chipped and the top end of the pipette is smooth and flat. If the top end of the pipette is chipped or not smooth, it is very difficult to pipette. Acclimatize the pipette. Acclimatizing the pipette means to rinse the pipette with the solution to be used. In this video, we will be using solution A. When using the pipette, do not pipette the solution directly from the flask in which the solution is stored. This could contaminate the solution. Always choose an appropriate clean and dry beaker and pour a small quantity into a labeled beaker. Here we have two beakers which contain the same volume of solution A. Compare the depth of the solution between the 50 ml beaker and the 250 ml beaker. The solution in the 50 ml beaker has more depth, and it will be easier to pipette the solution out of this beaker. To fill a pipette for acclimatization, use a rubber bulb to pull up the solution. If you're right-handed, depress the pipette bulb in the palm of your right hand. Hold the pipette in your left hand, but keep your index finger near the top of the pipette. This will make it easier to put your left index finger over the top of the pipette and hold the level of the solution when you remove the pipette bulb. Keep the tip of the pipette below the surface of the liquid to avoid sucking up air and sucking the solution into the pipette bulb. The level of the solution should always rise up the pipette. The solution should not drain back into the beaker. For acclimatization, we draw up a small amount of the solution, about 1/3 to 1/2 of the bulb of the pipette. Remove the bulb and tilt the pipette horizontally to rinse the inner walls of the pipette. Make sure to let the solution slide past the calibration mark near the top of the pipette and rotate the pipette to ensure the solution touches the inner walls of the pipette. Drain the solution completely. After draining all the solution used in the acclimatization, dry the top of the pipette thoroughly, and repeat this step 3 times to finish acclimatizing the pipette. Pipette and deliver 10.00 ml of solution A. Fill the pipette with the solution past the calibration mark. Remove the bulb and quickly place the index finger of the left hand over the top of the pipette to hold the level of the solution. Tilt the pipette slightly and wipe away any liquid on the outside surface. Using the same hand, place the pipette between the second and third finger to support the pipette and pick up the beaker to bring the pipette calibration mark to your eye level. Slowly release pressure on the index finger so that the bottom of the meniscus approaches the calibration mark at eye level. At the mark, apply pressure on the index finger to stop the level of the solution such that the bottom of the meniscus is sitting on the calibration mark. Inspect the tip of the pipette to make sure that it doesn't have a hanging drop. If there is a hanging drop at the tip of the pipette, touch the tip of the pipette on the wall of the beaker and recheck to ensure that the position of the meniscus has not changed. Now you're ready to deliver the solution into an Erlenmeyer flask. Bring the pipette to the Erlenmeyer flask and release the pressure on the index finger. Drain the solution into the Erlenmeyer flask with the pipette tip touching the wall of the flask. After draining, wait 10 seconds before removing the pipette. At this point, the calibrated amount of liquid has been transferred. Look closely at the tip of the pipette. A small portion of solution remains in the tip. Do not blow out the pipette.
So that's it. In this activity, we better understand how to properly use a pipette. And therefore, we conclude that we must use the pipette correctly in the laboratory in order to avoid mistakes, dangers, and accidents such as spilling any solutions in the future. So, thank you for watching.